Okay, so I think we're a couple minutes in. Um, as I mentioned, both talks today are about morphogens to control in vitro neural differentiation fates. And our first speaker is Soraya Scuderi. And so I'm glad you accepted the invite to come speak, and we're really excited to hear what you have to say. So you can share your screen. I think you'll just take it right away from me and go ahead. Thanks.
Thank you, Carlo, and thank you everyone for joining us today. I'm Soraya Scuderi from the Vaccarino's lab at Yale, and will present our work on the specification of new marine neural lineages using an orthogonal gradient of wind and sonic edging in organoids. As many of you in the audience, we have been interested in studying human brain development for a while by using stem cells and organoids. But I think one of the latest and old-fashioned question that we have in mind as a developmental biologist is how do embryonic tissue that are organized in space and times can give rise to all the lineages and specify cell type. And of course, behind this, there is a very old concept of the morphogen gradients, these soluble molecules that can travel long distance and inform the cells in a tissue to acquire a specific gene pattern of expression that then allow them to function. And here represented in a simple way by the French flag model of Woolpert. So with this in mind, we wanted to kind of create a better model of organoids in vitro and to really study the effect of the presence of multiple morphogens on the neural tube. And to do this project, we team up with a group of bioengineers at the bioengineer departments with the Levchenko lab. And we design a new mesofluidic device that I'm showing you here in the center that consists of a culture area and is surrounded by sources and sinks, in which in the source we can apply the morphogen of interest. To start our project, we focus on two main morphogens that are known to be responsible of the dorsal posteriorization of the neural tube with the molecules of wind. And the other morphogen that we wanted to model was the sonic edge that is known to be secreted by the notochord and responsible of the ventralization of the neural tube. On the right here, I'm showing you a zoom in of our culture area. This is very important to then when I will present our results. So the culture area consists of 5 consecutive chambers that are communicant. And so the flow can pass through the chamber between the source and the sink. And we apply the 2 orthogonal gradient so that we will have a region that is close to the source of wind and is indicated by the position C1, while the position C5 is the one that is the furthest from the source of wind. At the same time, we have a region in the culture area that is highly exposed to the morphogen of sonic hedgehog, while on the opposite side, the organoids that will acquire that position will receive less sonic hedgehog. We name this device as DuoMap, that stands for Dual Orthogonal Morphogen Assisted Patterning Specification. So first of all, we want to stress the concept that our device is just based on passive diffusion. So it means that the morphogen gradients is created just for the movement from a highly concentrated solution to a less concentrated solution. And is highly dependent on the molecular weight of the morphogens. So what we did, this is my colleague at the Levchenko lab, Tae-Young Kang, we tested and proved that we are able to create morphogen gradients by using fluorescent probes that have a similar molecular weight of the morphogen that we were going to apply in our true experiment. So if you follow me here with the green fluorescent probe, we tested the molecules that was going to activate the wind pathway. And on this line plot, we showed that measuring the intensity of the fluorescence, we could create a gradient that first was stable for several hours, up to 48 hours. The 3 lines colors indicate different time points, and that along our culture area, we will creating a different concentration of the morphogen with the C1, with the highest concentration of Wnt, and the C5, the least. And similarly, for the other morphogen, the big protein of 20 kilodalton sonic hedgehog, we use the true recombinant protein, we could have a zone in the culture area indicated with V that show high concentration of the morphogen opposed to the counterpart that we indicated as D. And the diffusion profiles and the gradient was also modelized. So we confirmed that, of course, the presence of a double orthogonal gradients, they influence slightly each other, but we still see a gradient formation as indicated by red, high concentration of the morphogen in C1. And while we proceed in the culture area, we have a reduction of the morphogen concentration. So with this basic principle, and once we prove we can form a morphogens gradient, we decided to apply to 7 independent iPSCs line. We form embryon bodies, and the neural induction actually occur in the mesofluidic device, in which the embryon bodies are loaded in the culture area through, if we follow my laser pointer, through this pinhole into an hydrogel. Once the hydrogel polymerizes, each of the embryon bodies will acquire a specific position in our culture area that is a kind of fixed for the entire patterning. The patterning lasts only 5 days, and at day 9, we perform our first collection time point. And it's very important that I guide you and I explain our sampling strategy. So what we wanted to keep for our follow-up analysis was the positional information within the culture area. And so we decided to do a subdivision of the culture area in 10 positions that we identify as first along the major axis in C1 to 5. And then we further subdivide when we were collecting the sample in sample belongings to the ventral region of C1 because exposed to more sonic hedgehog compared to the sample derived from the dorsal region. So we have in total 10 samples and we keep them in cultures in separate dishes for further analysis that we perform later on after 2 months in culture. But let me show you, so the first things right after the patterning at day 9, we, by bulk RNA sequencing at this early stage, we wanted to test the effect of the morphogens on our organoids that were occupying specific location in the device. And to do so, we are representing here in this gene expression relative level, known genes that are particularly enriched in primary versicle of the neural tube, while we move from the most anterior, like forebrain, to the most posterior, the hindbrain. And to convince you that our device, we achieve the patterning, if we look, for example, at the expression of a non-forebrain marker, like the FOXG1, the expression level of these genes was highly expressed in those embryon bodies that we recovered from the most, from the sample, from the position that was further from the source of Wnt. So and similarly, the highest expression was present in the low region. Of course, if we look also on the samples derived from the ventral position of the device, there was a similar trend. Because we know that the FOXG1 is also expressed, for example, in the subpallium. And another example is that the organoids respond properly to the morphogen with the specification of posterior region. We can follow genes as GBX2, the OXA2, and IRX3, that instead they were highly expressed in those samples that were occupying the position C1, that is the one close to the source of Wnt. And other example, we confirm also the dorsal ventral like fate acquisition by looking at several other genes. But so with this in mind, we achieve we think we achieve this change in pattern of gene expression after just 5 days of orthogonal exposure. We wanted to ask the following 3 questions. Which neuronal lineages we were able to give rise in these organoids? Which are the pattern of gene expression that control the lineages? And then, because we tested several lines, do this IPS line respond in a similar manner to the gradients? So to start to answer this question, we perform single-cell RNA sequencing at this late stage. And as you can see here, we are proposing a unique UMAP because all the cells coming from all the 7 iPSCs line were clustered together in a unique data set that then we process for annotation. And surprisingly, we were able to identify a variety of neuronal lineages. Spanning, for example, here I can show you from excitatory neurons from the cortex, inhibitory neurons from ganglionic eminence, hypothalamus, thalamus. Actually, pretty surprising also GABAergic neurons like the cerebellar Purkinje cells, and not least also midbrain dopaminergic neurons. So all of this was also confirmed by protein analysis. And taking advantage of the numerous human brain atlas that were coming out in the latest years, we also, through library transfer and projection score, we confirm the identity that we assign to the main branches of lineages that you can appreciate here on the UMAP on the left. And this was, for example, the Brown et al. Human brain atlas, that they dissect all these regions that we are listing here, spanning a very important stage for our study because of the first trimester. So this kind of reassure us, and it's very impressionant how the organoids at certain level can model actual human brain samples. And not least, we wanted also to, of course, we use other tools available like the Voxant projection, in which we project our different cluster in, for example, an E55 mouse structure, and we saw an enrichment in those structures for the transcript correctly associated to the lineages. But our next step, we are interested in the pattern on the role of the morphogen exposure. So the first analysis that we want to do, we are showing here an average cell composition. When we want to dissect the positional influence of this morphogen, the first striking feature that we saw was that non-cluster like the ganglionic eminence interneurons or the hypothalamus in light blue, and instead the ganglionic eminence in red, they were produce this lineage, particularly in those samples that were exposed to the highest concentration of the sonic hedgehog. So this was the major differences, while on the other parts, on the low sonic hedgehog region, we were observing, besides the cortical excitatory neurons, the cerebellar Purkinje cells. So of course, we want to add a statistical analysis to this observation of cell proportion. And so thanks to the help, all the analysis on single cell have been performed by Alexandre Jourdan. And we perform this linear model in which we could really classify the influence of each morphogen with the prevalence of a cell lineage. And so within the position in the device. So to summarize here, we saw that under the influence of sonic hedgehog, we have a higher proportion of, for example, the inhibitory neurons I was pointing before, or the midbrain ventral progenitors. Instead, if we want to dissect the influence of wind, and we look at this as more a slope while we move through the chamber position, we can see that there is an anti-correlate proportion while we increase the concentration of wind. instead for the excitatory lineages. Instead, for the midbrain ventral progenitors, it seems that this lineage needs the influence of both the wind and the sonic hedgehog. So then we put all together this single analysis to have a recap of the influence of the morphogen on the different lineages. And so you can identify, we narrow down per each lineages which was the best condition of morphogen concentration that give rise to the lineage. And so if you see indicated just an S, means that this lineage actually is, in this case, is more abundant in absent in the low sonic hedgehog region, while we have other lineages that are still under the influence of sonic hedgehog, but actually they are more abundant when we have more concentration of the sonic hedgehog. But more than this, once we tested and saw that the different lineages were they were influenced by the morphogen, we wanted to go back at the early stage to dissect if the exposure to the morphogen was able to activate specific pattern of expression. And we call this morphogen response spatial modules. Because as you can see, like this is a miniaturized schematic of our culture area. And what is plotted in is we apply a k-means of 20. So we identify 20 gene patterns that follow, they are influenced by the morphogen. And so they are highly expressed or in a position respect to another. So they must be differentially expressed between the position within the chambers. And so we could identify, we then put in relationship this pattern of expression and correlate them with the probability of generating the lineages at the later stage. And so we could identify some pattern that univocally like were mostly associated, for example, with the excitatory neurons, that is our blue cluster here, for highlighted that is specified early on by correlate well with the pattern 2. And so then if we go and look at these genes, we again find, for example, FOXG1, TBR1. And we also investigate the presence of this pattern directly in the true fetal brain atlas in the progenitors' lineages to see if also in that case there were predictors of the different dissected regions that are listed here on the bottom. And so if we focus again on the pattern 2 that we saw predicted our excitatory lineages, it was also highly strongly correlated with the cortex hippocampus progenitors cells in the true human brain. So then we move on and we wanted to check the line-to-line variability, because overall, all the individuals were able, as you can see here on this hierarchical clustering, to generate all the variety of lineage that we capture at 2 months old by the single cell. But then when we look more in details, position by position, is there that the variability line to line arise. So here I'm showing you, for example, these are our 6 lines that we perform we perform the single cell. And you can see that we have an individuals, the 1, 1 to 0, that generate the list of, for example, the excitatory neurons lineages. While, and similarly, like, for example, in the position C1V, these individuals generate more hypothalamic neurons compared to others. And so here it's like we want to confirm at the protein level by monocytochemistry, for example, this variation in cell proportion among individuals. And here I'm showing the dopaminergic neurons by label bar TH between the 2 individuals that I highlight with the dots. So really this variation was visible. But so we wanted to went back because to the early stage, because something must be different in these lines when they were patterned in the device and responding to these morphogens. And so we thought to take advantage of the morphogen response spatial modules that we created to identify line-to-line differences. And speaking about, so here are our pattern of expression. And in this line plot, each line that is different colors indicate different individuals. First of all, what we observe is that if we compare the organoids obtained from the ventral position, the one exposed to isonic hedgehog, overall the lines show a similar pattern of expression, like response of this pattern. While in the dorsal derived samples, we saw like that is the most influenced by the wind, because in this case there is less exposure to sonic hedgehog. They were quite variable. And this was also statistically tested with the standard deviation. You can see an enrichment in dark dots that are sample derived from the dorsal position in the device. So we wanted, again, to confirm if this early pattern resolve at the later stage in a overproduction of the lineages, so to put together basically early and late events. And what we observe is, let me give you the example of our pattern 2. We have these two lines that are like 07 and 01 followed by RDH, that in the dorsal position show a different trend compared to the other 3 lines. And this, again, this pattern consists in the activation of FOGG1, TB1 in the position that are further from the wind. And this later on, by single cell, we show that as a result, an increase in the production of the cells coming from the forebrain that is indicated by this light beige in our pie chart. But of course, line-to-line was also true, or I can give you another example of a pattern that is quite active in the most posterior region. If you focus on the individual 1120 with this line orange, he separates himself in terms of a pattern of expression compared to the rest of our iPSC's lines. And this pattern actually are responsible of the specification of hindbrain region, like IRX, PAX3, more posterior region. And this again was revealed also later on, as you can see by the increase in the expression of lineages belongs to the hindbrain. And we show a similar, like, of course, we wanted to also analyze the samples derived from the ventral region, and we saw some difference in the propensity of the lines also to respond to sonic hedgehog itself. For example, we identified these 2 individuals, the dark blue and the light blue, that they seem hypersensitive to sonic hedgehog, and this was also proved by the percentage of striatal or hypothalamic neurons by single cell at the later stage. So I hope I show you enough that just 5 days of the patterning in our duomaps is able to generate a variety of neuronal lineages, that we identify important spatial modules of gene expression that are activated very early on, but they reflect neuronal lineages later. We believe that our system is versatile, because we can model different morphogen, combination, timing, and concentration can be modulated. So now we are like, in our future direction, we want to study like iPSC's diversity to be in the topic of the talk. And so we want to extend this study to an increased number of lines, repeating the same line, add more clones, because we want to understand like what are the differences that are driving this response to the morphogens. And with this, I want to thank you all the Vaccarino Lab members, in particular Alexandra, then of course the Banjanier team with Levchenko and Lab and Inte Jung Kang, and the Habits of Life at Mayo Clinic. And of course, we thank all our fundings to support us our research. Thank you. I'm open to questions. Thank you.
Thank you so much, Soraya. Let me see if, just jump in, no need to raise hands or anything, guys. I think we're a small enough group. But it's great to see you already using multiple lines in your first steps of exploring your morphogen gradient setup. Have you thought about, as you begin to think about diversity across lines, in addition to watching how they respond differentially to the gradients and to the morphogens, have you thought about or collected any information on their starting point? So do these lines look different coming in at the stage of, I can't remember exactly what it was, the embryonic body that you drop into that chamber?
Yeah. So actually, yes, we collect bulk RNA sequencing at the iPSC stage. So we are looking into this data. There is a kind of a strong signature of batch sequencing that we are trying to correct. But yes, that we are really we thought about it. So luckily we collect
That's wonderful.
the IPS sample at the right time. And the truth is at the beginning, because this experiment was done in 2 batches. So the first 3 lines, and in the second experiment, we had the 4 lines. Honestly, with the first batch, we didn't see much of variabilities. But of course, the more you increase the N, when then we put together the data, the variabilities start to arise. And yeah, we, of course, the device has been over time troubleshoot before to reach the actual design that you see today. And so several times the same line was repeated, and it seems that there is kind of really the same propensity, but we want to repeat the experiment to confirm.
Very cool.
So I see that we have some question in the chat.
Great, go for it.
Lines running independent device. Okay, so first question. As you can see, of course, the device is relatively small, so can allocate 200 embryo bodies. But then, because with our sampling strategy, actually we perform per each line, we load at least 5 separate chambers, so 5 devices. Because our lines run in independent device. Yes, we do independent device and multiple device per individuals.
And a male-female question. Yeah.
And then, yes, we are also planning to apply on male females, but we didn't do yet. But we are thinking about, because we are also interested in sex differences. Thank you.
I wonder if I could ask you a technical question about the last, your slides 10 and 11. You seem to be showing consistent differences between the dorsal and ventral regions of the chip, of the microfluidic device. Is that right? And at some point you were showing that. And it made me wonder whether when you do the analysis of line-to-line variation, is that dominated by cells that were grown in that dorsal domain? Yeah, that's the question.
No, please. Yeah, so for our main analysis, we put together all the samples. So we did this average cell composition, but then we look line to line, and we start to observe a different propensity of the line per position. Yes, and
let me rephrase the question. Yeah, here we go. So this in the middle here, the position is the inferred position in the brain, but on the right-hand side is the position in your chip. Isn't that right? Am I making sense? I think this is quite an interesting question, actually.
Are you referring to which panel? The one in the
here?
center?
So in the middle now, you've got this blue and the red, for example, is dorsiventral telencephalon.
Yes.
And there's clear differences between the lines. Great. So isn't that right? But then on the right-hand side, that dorsal and ventral refers to the position in the chip. Isn't that right? Exactly.
Yes. So let me show. They are listed here, the various positions on the bottom. Here I'm giving you just a flavor, like only 3 positions, basically. But here on the right, we are looking at the entire position and the entire culture area. So we have the 5 positions coming from the dorsal and the 5 position sample coming from the ventral area in the culture.
Precisely. So what I want to just ask you now, I couldn't ask you really quite precisely. The variability is there's much more variability in the dorsal.
Yes.
In the dorsal data than in the ventral data, and I'm wondering why that is. And one possibility, it seems to me, is that, I mean, I think that's potentially very, very interesting. But one possibility is that it's just simply a reflection of the growth rates. So there's an ascertainment bias that you have more cells in D than in V in that state. So if you corrected for that, then this variability would increase, or the other way around. I mean, do you see what I'm saying? Are they growing the same way in the dorsal and ventral domains?
Actually, the ventral domains seems they are slightly bigger. In the ventral, that is the high sonic edge of exposure. So, but of course, it's a good question. We might want to look to exclude that is kind of a growth rate. But
maybe we could just extend the idea a bit, because early on, you know, you were showing us images where if you could keep the cells under the embryonic bodies under constant observation, you could almost monitor by imaging the precise periods of growth. Anyway, I thought this was extremely interesting to me. The differences that you showed here. Thanks so much.
Yeah, thank you. Yeah, we think also that is basically interesting, and also is revealing that maybe the level of wind that we are applying is not sufficient to instruct in a strong way all the lines. But this difference can be assessed only if we replicate and also maybe test a stronger concentration of the morphogen. Thank you.